Good morning, my dear students. Today we will discuss about synthetic seed technology. Plants are generally propagated mostly through seeds. In some crops, the use of seeds for propagation has not achieved success because of seed heterozygosity, minute size, absence of endosperm, and absolute necessity of fungal infection for germination. Vegetative propagation can be employed for multiplication of some of the plants. However, conventional vegetative modes of propagation are time-consuming, expensive, and cannot produce plants at larger scale. Production of artificial seeds using synthetic seed technology can play an important role as alternative to other conventional methods for large-scale propagation of useful crop varieties. Artificial or synthetic seeds are somatic embryos, shoot buds, nodal segments, or any other plant tissues encapsulated by substances like sodium alginate, agar, gel-rite, and sodium pactate, which can be used for sowing as seeds and possess the ability to convert into plants under in vitro and ex vitro conditions. The naked somatic embryos or shoot buds, when exposed to natural environment, will not survive as they are highly sensitive to desiccation and other infectious pathogens. They are therefore required to be coated with protective covering. Apart from giving protection, the encapsulating agents should also provide nutrition to the developing embryos. Now let us come to the history of synthetic seed development. The first synthetic seeds were introduced by Keto and Zenick in 1982 when they encapsulated carrot somatic embryos with polyoxyethylene, followed by the desiccation. The somatic embryos and calyces of carrot were enclosed in polyoxyethylene glycol, which was allowed to dry for several hours on telephone surface under the laminar flow cabinet. The dried mixture was inoculated onto freshly prepared medium, which was then allowed to rehydrate. The polyoxyethylene was favorable for use as coating materials for synthetic seeds as they were non-toxic, readily soluble in water, and can be easily dried to form thin film and do not support the growth of microorganisms. Redenbach and his co-workers in 1984 developed a technique for encapsulation of single hydrated somatic embryos of alfalfa plant. Since then, encapsulation of somatic embryos of several plant species in hydrogel have been successfully performed by many workers. Let us discuss about the synthetic seed production. The production of synthetic seeds was primarily based on the encapsulation of somatic embryos before it was extended to other plant tissues generated under in vitro culture condition. The somatic embryos are developed from somatic cells in vitro, unlike zygotic embryos, which are obtained from fusion of male and female gametes. The normal zygotic embryos have protected seed coat and endosperms, which provide nutrition to the developing embryos. The somatic embryos, on the other hand, lack both endosperm and protective covering. So, generally, The naked somatic embryos are enclosed in protective coating materials to produce synthetic seeds. The coating materials, however, should possess the following characters. It should not produce damaging effect to the embryos. It should be mild enough to allow germination, but it should also be sufficiently durable for rough handling during manufacture, storage, transportation, and planting. The protective coat should not only give protection but also provide nutrition to the embryos inside for germination, and the synthetic seeds should be transplantable using existing farm machineries. Several gels like agar, alginate, carboxymethylene cellulose, gelrite, gorgum, sodium pactate, etc. have been tested for encapsulation of somatic embryos for synthetic seed development. However, alginate encapsulation was found to be most favorable for providing protective covering to somatic embryos. This is because the alginate enhances capsule formation and provides sufficient firmness to bits containing somatic embryos to withstand mechanical injury. The encapsulating matrix is provided with nutrients and other growth regulators which would play the role of artificial endosperm. The incorporation of nutrients and growth regulators to the encapsulating matrix results in the increase in efficiency of seed germination and viability of somatic embryos inside it. Other adjuvants like fungicides, pesticides, antibiotics, and microorganisms, for example, rhizobia, may be included into the encapsulation matrix to instill different properties to synthetic seeds to suit to prevailing conditions. Now, let us come to different types of synthetic seeds. Two types of synthetic seeds are produced following different approach of development. They are desiccated and hydrated synthetic seeds. Now let us first take up desiccated synthetic seeds. In this case, the somatic embryos are first encapsulated by polyoxyethylene and allowed to desiccate. The desiccated synthetic seed can be developed for those plant species which have somatic embryos resistance against desiccation. The carrot somatic embryos can be encapsulated with polyox by mixing equal volume of embryonic suspension and a 5% solution of polyox to give a final concentration of 2.5 polyox. The suspension was dispensed as 0.2 ml drops from the pipette onto the telephone sheets and dried to wafer in a laminar flow hood for around 5 hours. The embryo survival and conversion of seeds are determined by redissolving the wafers in freshly prepared embryogenic medium and culturing the rehydrated embryos. The synthetic seeds with desiccation to 10 to 15% can be stored at room temperature for about 1 year without being lost in cell viability and germination potential of the seeds. Now let us come to hydrated synthetic seeds. The hydrated artificial seeds consist of somatic embryos or suitable plant tissues enclosed by a hydrogel. The substances like potassium alginate, agar, gel-rite, sodium pactate have been tested, but sodium alginate was found to be most effective coating material for hydrated synthetic seeds. For the production of hydrated seeds, the plant materials are mixed with sodium alginate gel 0.5 to 5%, which is followed by dropping into the calcium chloride solution about 30 to 100 millimolar using pipette. Round and firm bits of calcium alginate containing the somatic embryos are formed as the ion exchange occurs, resulting into the replacement of sodium ions with calcium ions. The hardness and rigidity of capsules mainly depends upon the number of sodium ions exchanged with calcium ions. In most cases, 2% of sodium alginate gel when complexed with 100 millimolar calcium chloride solution produce desirable quality synthetic seeds for many plant species. We will take off the propagules used to produce synthetic seeds. Various micropropagules such as somatic embryos, shoot buds, embryonic masses, and protocome-like bodies have been used for synthetic seed production. First, let us take up somatic embryos. The somatic embryos are the most favorable propagules used for production of synthetic seeds. Artificial seeds using somatic embryos have been successfully developed for a number of important crops. The somatic embryos possesses radicle and plumule, which will develop into roots and shoots respectively in single step without subjecting to any specific treatment. With the advancement of plant tissue culture technology, somatic embryos have been induced successfully in number of plants. This makes somatic embryos more preferable for artificial seed production as they can be more easily available. When the somatic embryos after encapsulation were dried to 10 to 15% and stored in dry state for about 4 weeks, 65% of the somatic embryos survived and germinated like the true seeds. Now let us come to apical shoot tips or shoot buds. The unipolar shoot buds or apical shoot tips, which do not have the root meristem, have also been used for production of synthetic seeds. These propagules, as they do not have the root meristem, have to be induced to produce rooting before they are encapsulated. The shoot buds of apple clonal rootstock M26 have been encapsulated after rooting induction by treatment with IBA for 3 to 6 days. 85% conversion of the synthetic seeds was observed when apical microcuttings of M26 apple rootstocks was encapsulated after treating them in 24.6 micromolar IBA. Now we will take up protocomes and protocome-like bodies. When orchid seeds are inoculated on the medium in vitro, the seeds germinated and form spherical bodies called protocombs, which can be differentiated into complete seedlings with leaves and roots. In different orchids like Cymbidium zygentium and Dendrobium wardianum, synthetic seeds have been produced by encapsulating the protocomb or protocomb-like bodies with sodium alginate gel. Now, let us take off application of synthetic seeds. The artificial seed technology can be used for multiplication of economically important crops, ornamental hybrids, and rare medicinal plants which are on the verge of extinction. Many of these plant species cannot be propagated vegetatively or produce less seeds with low germination frequency in nature. Artificial seed technology can be alternative for mass propagation of these plants. There are several potential uses of synthetic seeds for those crops such as citrus, grapes, mango, etc., which are vegetatively propagated and have long juvenile periods. Production of synthetic seeds in medicinal plants by encapsulating the somatic embryos and other vegetative propagules such as shoot tip and nodal segments have solved some of the problems involved with the propagation and preservation of plants with medicinal values. One of the advantages of synthetic seed technology is that it offers space and equipment saving option for storage of plant materials of different rare and commercially important plants at low temperature. Another application of synthetic seeds is that they are easy to handle because of smaller size bits, have higher scale of capacity, possibility of automation of the whole production process, and direct delivery to the field. The artificial seeds with vegetative propagules enclosed within them can be used for exchange of exogenic plant materials between the laboratories. The transport of synthetic seeds between the countries does not require quarantine department permission. The synthetic seed method also has potential for long-term storage in liquid nitrogen, which is a process called cryopreservation, and also for short-term storage without losing the viability of the seeds. Another important positive point of artificial seed technology is that they are independent of seasonal variations, as they can be produced in the controlled laboratory environments. There are limitations of synthetic seeds. The first one, production of good quality micropropagules in high number is prerequisite to production of synthetic seeds. However, there are limitations in generation of viable micropropagules which are useful in artificial seed production. Second point, though somatic embryogenesis is successfully induced in number of plants. Very often, asynchronous development of embryos is observed, which limits the production of normal plants after science seed production. And third point, improper maturation of somatic embryos, which reduces the success rate of conversion of synthetic seed into normal plants. Fourth point, lack of dormancy and stress tolerance in somatic embryos limit the storage of synthetic seed. Lastly, the conversion of synthetic seed into plants successfully is the most important aspect of synthetic seed technology. Due to various reasons, the conversion rate is always very low, which limits the commercial application of this useful technology. Now finally, let us come to the conclusion part. The artificial seed technology is considered as an alternative to conventional plant propagation methods by generating several important plants rapidly in large scale. It also combines the advantages of clonal multiplication with those of seed propagation and storage. Artificial seeds also offer tremendous potential in micropropagation and germplasm conservation of rare and economically important plants. However, the lack of production of high quality micropropagals for synthetic seed production, coupled with low conversion rate to normal plants, has restricted the widespread use of synthetic seed technology. Further, detailed investigations are required to refine this useful technology for its application at commercial scale.